last time I present, of course, it's some kind of my story regarding space medicine. Today, of course, space bioscience is much broader, actually, field of research, which include just biology, new technological developments, molecular biology, biotechnology, whatever related to actually potentially to space exploration. And the principal mission, of course, of the our branch, BioSciences branch at NASA Ames Research Center, is advanced space exploration by achieving new scientific discoveries and fundamental discoveries as well, and technological development in biosciences. Yeah. Space life science, yeah, of course, it just includes many categories, yeah, and it's comprised field of science that involve all fundamental scientific research on living organisms such as microorganisms, plants, humans, being, yeah, and well, we know that actually two major components which probably provide more information for us, and we would like to know it's how living species respond to actually a space environment. Can they survive in this environment? Second one, it's origin of evolution of life in the universe. Of course, it's very exciting area because we want to know whether life can exist and whether it exists and in which form beyond the Earth. And this slide actually shows that without any exception, we know that life on Earth in the sphere is carbon-based, right? And it's organized with a phase boundary of membranes that envelope reactive biomolecules. That's what we call cell, right? Every actually probably documented terrestrial life is self-replicating entity that has genetic information which we know it's form of DNA or RNA, right? If any another extraterrestrial biological system exists, the modes, information storage, processing, and reproduction should be or should maybe not identical to what we have on Earth. Well, understanding of this prebiotic evolution, of course, and search for extraterrestrial life somewhere in the universe, it's a major goal of our program, astrobiological program at NASA Ames Research Center. We have Institute of Astrobiology. So we know that what is life actually, and if we just kind of first of all, it should be, we believe, life originated in presence of liquid water. This is why just we're looking and Kepler looking for planets which would be similar to Earth, at least to contain water. Of course, it should be a reasonable source of usable energy, which is necessary. And general criteria of life, you absolutely aware. It's of course, first of all, it should be something movement. It should be organized in some kind of species. It should be sustainable. It should remain homeostasis, ability to maintain constant conditions within the body. Of course, it should absorb and use energy, and of course, reproduction. And the third one is the last one is growing in development of some organs, a new type of cell, and so on. That's why it's so important, because people just kind of exciting to look something which probably would be similar to actually to find life in the universe. So why we just look in this? You remember probably at NASA Discovery in 2010, when suddenly it was found some organisms, potentially a strain of bacteria living in California lake, which potentially should build DNA from the incorporated, not phosphate, but arsenate. And that was kind of very exciting discovery, and research team which discovered this bacteria actually suggests that these bacteria incorporate arsenic into DNA in place of phosphorus, which essentially kind of gives us completely different actually life, arsenium-based life, because we actually know that in all life we're using ATP and oxidative phosphorylation. So, but later it turns out that unfortunately this experiment was actually not corrected, and that was kind of disappointment that we were not able to prove that could be another form of life. Yeah, another exciting actually discovery, which probably you know, it created huge noise in community, scientific community, when in 1996, David McKay from NASA Ames Research Center actually made picture of Martian meteorite, which they found and was under this number, which presented actually on the picture. Well, of course, scientists at first moment just kind of consider this image that it's something like bacteria-like image, which you can see in the center, and they kind of was excited about discussion that it's possible extraterrestrial life, and of course, question is that life is survived in meteorite when it's arrived to Earth through the atmosphere, and this actually. Well, there is no, of course, official NASA position about this meteorite. NASA scientists can kind of discuss any hypothesis, so we know this, yeah. But point is that it's raises a question about survival in open space, which actually become a common issue for bacteria. You remember probably that story about Apollo 14 camera, which was left on a moon surface. And 1 year later, it was picked up, and they found that bacteria, Streptococcus, I guess, yeah, survived perfectly under these conditions in this space. Yeah, and of course, what we know, if it's true, if it's possible that meteorite or life can arrive to the Earth from the space, from another planet, it just, of course, actually prove maybe a hypothesis of panspermia. Of course, it's maybe unlikely to accept this hypothesis at this point, but it still make sense. And on the previous slide, I just kind of demonstrate and show one creature which we all kind of exciting because it's unbelievably resistant to space flight environment. This is tardigrade, or in other words, the water bear. It's aquatic, very small animal, which typically grows to probably like 1.5 millimeter of length. But what's remarkable about this animal that it can survive in liquid nitrogen, and it could be heated to 150 degrees. And nothing happened. It can actually withstand huge pressure, like 6,000 times more than human can survive. And most important, it's very, very resistant to radiation. I forgot, it was probably 2010 when astronauts placed outside of International Space Station some container which contains such species. And it's remarkably, they survive in a vacuum and on the space radiation. Why it's important? Because we believe that actually it should be some special kind of condition, or when animals and microbial species can survive. Well, in my laboratory, we just currently running research on metabolic control, so-called project, when we actually trying to reduce metabolism of animals to the minimum level, like 98%. And in this state, yeah, animals becomes very resistant to radiation. They can survive conditions, environmental conditions, which otherwise would be lethal. What we believe, and actually this experiment with the water bear shows us that it survives not just in a normal state, it survives in a dry state. In this state, yeah, they actually get to the stasis, hypermetabolic stasis. So they can be like 100 years in this stasis. So we believe that all microbial actually life in somewhere in another planet could be also in some kind of hypermetabolic stasis. We have some kind of hypothesis, of course, yeah, which demonstrate importance of this reduction of metabolism for survival. For example, in this plot, you can see adaptation actually using two strategies. And blue line shows you how environmental conditions can change over time. And the green line shows that organisms, of course, trying to adapt through the mutations to these conditions. This is one possibility. But sometimes variation could be so significant that no any mutation can actually adapt animals or living creature to changes of this environment, and it becomes lethal. So one possibility which addressed here in the red line, that if it would be possible to reduce metabolism and get to some dormancy, which is well known in many animals and microbial species on the Earth, they can survive these changes in the environment until another better condition will come. So, of course, it's just kind of only a hypothesis, but we believe that, like presented in this slide, if any life in universe exists, it might be actually under some kind of in special state, which reminds metabolic stasis. By the way, you know that, for example, in the Earth, 80% of Earth's biosphere, it's permanently, its temperature approximately 5 degrees, right? And it's most important that this 80% of biosphere is microbial. Remarkably, that majority of this microbial population, like 99%, they are not in active state, but they call some kind of stasis. They're not culturable, most of them, and typically they demonstrate very low level of metabolic activity, but could be resuscitated if they become cultural. But most important, 99% of biomass or microbial population is in some kind of stasis. Well, we believe that in some kind of during evolution, sometime conditions change so dramatically that this microbial life must to survive only by reducing their metabolism, and they still remains at that state now. Of course, it's only a hypothesis, but we believe that it might be very general, and ability to significantly reduce biochemical metabolism and enter to dormancy or some kind of a metabolic stasis. It would give enormous advantage for different species to survive and preserve their genomic integrity, which is most important when the environment becomes threatening for their life. I would like, of course, we in our solar system, we have many planets actually which potentially could be suitable for life similar on Earth. And like I put this message here, if any life beyond Earth exists, it's likely to be at low temperature, and at low temperature, of course, it's very low metabolism. And we believe that conditions in Europe, for example, could be potentially suitable for such water bear, which we actually just discussed. And most important, scientists believe that there is some kind of liquid water on the ice in Europa, and at low temperature. What is important also that ice probably potentially would be very good shielding for radiation protection from space radiation. And so people even just kind of calculate that 20 meters of ice would completely actually reduce space radiation to what we have on the Earth on the normal ground level. So it's hope, of course, that we will probably find life beyond our Earth. But like I said, my impression that it should be probably in some special metabolic stasis. Now, if we return actually to the second point, how life responds to space environment, which is most important for us because we're just trying to colonize space. Actually, we should just kind of probably bring to attention importance of our International Space Station, which is unique, actually, possibility for us to access to space. And we know how many very exciting breakthrough experiments now currently actually we have during all these 2 decades mission in International Space Station. And you know that in 2005, for example, NASA Authorization Act designed an American segment, of course, because it's Russian, Japanese, American, to the International Space Center or International Space Laboratory, which gives a very exceptional status actually for research. Of course, it's first what we probably appreciate from International Space Station. It's kind of continuous exposure, very prolonged exposure to low gravity. This unique experiment we cannot actually afford and design on Earth. In spite of this, we have a lot of new technology which probably allow us to mimic low gravity, but of course we will never actually have possibility to actually fit real low gravity. So last time I just discussed that most of research on International Space Station, of course, focus on human research. In spite of that, a lot of cell cultures and even animals, we're trying to kind of design experiments with future flights. One, of course, a very simple, probably question: how cells, just individual cells, respond to low gravity? What's going on actually in this? And not much we at this point can actually discuss, because it's not sufficient data. In this slide, I present only you one actually call, which is unusual, because we all know that actually cells really change under microgravity, and here just it changed not only fluid distribution, gene expression, signal transduction, and all metabolic reaction. Well, in this slide I will show just that now just it's people exciting that whatever we will design our long-term mission to another planet, probably we will take advantage of innovative approach which medicine now using based on stem cell properties. And this actually gives you a slide about request for proposal which related to NASA mission involves stem cell research, because as you know, just the federal government until some time was against of such kind of research, and now we have green street to investigate stem cell in space environment, because they're important, you know, it's for different kind of therapeutic treatment actually, which is very important therapeutic drugs. Now they can restore, for example, heart tissue during heart attack. It can restore narrow, probably spinal cord actually activity during some kind of trauma or disease. So it's I believe it's one of the important actually issue which now we can address. We know that microbial cells, of course, also show some kind of physical changes in space, but these physical changes different from the actually stem cell, for example, which we have. And next slide, I will specifically present to you response of some microbial cell to low gravity. This plot demonstrates growth of cell and the low gravity, 0 gravity, and 1G, and you can see, of course, that increase in growth rate of bacteria, in this case Bacillus subtilis, compared to ground control. And what it tells us that bacteria cells, if they grow, they probably have higher pathogenicity. And indeed, some last experiment on International Space Station demonstrate that some pathogenic bacteria increase 2.5 times, even more, their pathogenicity compared to ground control after being in the space environment around like 3 months. How it's important of that? Of course, we know that if infection, bacteria infection somehow will raise, like Staphylococcus infection, or E. coli, or yeast infection, or fungal infection, it will be more resistant to antibiotic or any other antibacterial drugs. Next slide demonstrates changes, for example, in tolerance of bacteria to Disiptol. It's an antibacterial drug, and you can see that actually after 24 months, it's 2 years, they become more pathogenic and they become resistant to antibiotics. It's a very important issue which actually concern for astronauts because like 2 years or 3 years ago, they did not know that pathogenicity of bacteria increased. Why it's important? Because I will present this slide. It shows data from the bacterial contamination and fungi contamination on Mir station, Russian station, which is old and now just in an ocean. But you can see now that bacterial and fungi actually start to destroy all communications device over here. It just kind of even material biodegradation as presented in the left for rubber and even titanium. And most important, it's not bacteria themselves. It's products of the metabolism, in fact, this. Next slide, for example, demonstrate electrical tape and aluminum. You can see colony and products of metabolic which really actually cause material degradation. And this slide shows, for example, aluminum. You understand, it's very, very dangerous because it's very difficult to control microbial environment and the pathogenicity actually even in air of International Space Station. You remember probably 2 months ago it was cargo from Earth for International Space Station, and they actually postponed delivery because they found that there is some kind of contamination in this case. So it's important, of course, to address how cells, how bacteria respond to low gravity environment. In this slide, I present that our another platform which we using, and this actually very exciting opportunity. It's already 2 decades when animal research, cell culture research was employed in satellites. In this case, in May 24, last mission was BioSciences, and you're probably familiar about this. Our branch and our center participate in this with Russian Institute of Biomedical Problems. In this kind of slide, you can see on the top right corner group of researchers which involve in afterflight and tissue share, and Rich Boyle, one of the our researcher in the center. That was very exciting flight, of course. Yeah, it was I guess 46 animals sent over there. Let me just kind of confirm because I don't remember. Yes, that was 45 laboratory mice, and it was geckos, and it was snails and Mongolian gerbils because they're more resistant kind of to environmental changes. Unfortunately, payload experienced failure, of course, in flight. You know, probably heard about that, leading to death of 8 Mongolian gerbils. We know that all geckos and snails survive, and about half of mice, 45 mice, yeah, was also unfortunately died because of failure in the food kind of delivery. Well, last version which I heard 1 week ago from Russians because is that somehow mice get out from the container and cut electrical wire which provides some kind of food delivery to the chamber. Of course, it's kind of people always believe this was they sent actually 45 mice, assuming already probably that they not all survive the space environment. Probably it would be reasonable question. If now we have humans in the International Space Station, why we need to still send animals in space? And the answer is obvious. First of all, there are some experiments which we cannot perform on humans. Yeah, it's like biopsy or take a very invasive procedure which we can do with animals, but we cannot do with astronauts. Yeah, and second to that, that some experiment could be potentially dangerous for astronauts because in this environment we cannot control some reaction and some process which I mentioned about, for example, microbial contamination. So our division, of course, involved in this actually mission. several people, I guess, Rich Boyle, Eduardo Almedo, actually from our branch, and this is actually our new branch website. You can visit it in Ames Research Center, which actually describe our involvement in this research. And briefly, just only cover actually several most actually probably visible research in our branch. One of them is system biology, bioinformatics, leading by Victor Stoltz, who is just director of genomic facility at our center. We have kind of very comprehensive array actually facility. He can just probably he already involved and published numbers of very good papers in Science and Nature, focus on whole genome analysis. Now he's just involved in a little bit different area research when he's trying to estimate the effect of electromagnetic field on living organisms. And last year we published with him a paper which actually demonstrate actually very interesting phenomena that whole genome is kind of vibrated, it's cycling. So, and he believes, his hypothesis is that some magnetic radiation actually may significantly affect physiology. And this actually proved not only actually astronauts in International Space Station, but also in submarines, when it's also insulated environment, insulated from electromagnetic field. Next research, of course, is with Richard Boyle, and mostly focus on vestibular and nervous system. And he was one of the lead researchers which participate in the BioSciences mission. They sent snails to space, and human in a very similar to what they have in snails. And he using this model to actually identify some very important fundamental process. Next researcher is Sharmila Bhattacharya, who just kind of involved in research in kind of mostly immunology and oxidative stress research. Using her model is, of course, Drosophila. Drosophila, some genes and response to environment potentially could be a good model to mimic what we have in humans. My research involved in this kinds of metabolic control. You know that we just kind of already capable to put animals in very deep stasis, reducing their metabolism like 98% under these conditions. Animals like not alive, but this process is completely reversible. We can reduce dramatically like heartbeat from 170 beats per minute to 4 beats per minute actually, and it's reversible. It's very important, and we're doing this on non-hibernated animals, especially to demonstrate that potentially we can actually use this strategy for all animals. And of course we would like to switch to human-sized animals like dogs and pigs to demonstrate that we can intentionally induce stasis or reduce metabolism actually in these animals. Of course, you understand it's important for long-term missions, because we know now just, and I mentioned this in my previous presentation, that actually it's not probably possible to survive, for example, radiation mission. Payload's very expensive, and a lot of medical actually conditions and even just urgent conditions can arise during trip. So we believe that incorporating this kind of platform, strategy in space flight architecture will help actually to reduce impact of environment and handle some emergency life-threatening situations. Next research, of course, it's about radiation. We have Thor Strom, who is a radiologist. Now they involved in design of dosimeters, different kind of biodosimeters and dosimeter, just physical dosimeter, which actually allow to measure different kinds of radiation, like neutrons, space, high-Z radiation, proton radiation, and they try to design some universal actually device which would allow in lifetime to monitor such kind of radiation as presented on the picture. Another, of course, very important actually research in laboratory Rus Globus, Dr. Luz Globus involved in actually skeletal health study. She have probably like over the last 4 years a number of very good publications which used modern technology to evaluate resistance or changes in bone in response to space environment. Now I would like to actually switch to one thing which you probably already familiar because John Converse visited you last summer. It's about synthetic biology. It's a new initiative of Ames Research Center, yeah, which we believe would significantly contribute to actually our vision how we can modify or let's say customize some biological some lives in response to space environment for purpose of, for example, colonization or adaptation to environment on another planet. So I would like to share with you a couple experiments and couple ideas which John certainly did not mention because that we're doing in our branch. And first one, it's about probiotics. What's important here? You know that payload, it's very expensive, especially for long-term mission. Just water would be tons of foods. And if we come into drugs, for example, that in only intravenous injection liquid or water, distilled water for intravenous injection, for example, would require like 100 liters. So it's not possible actually to afford. Of course, a strategy for Mars exploration can include that we first have cargo ship which drop something on the Mars surface, and then astronauts will come. But also another idea, what if we would use synthetic biology to design actually symbiotic delivery of some products. We know that probiotics, for example, it's kind of friendly bacteria in our organism. In adult, it can reach like 4 kilograms per person in our intestine. It's very significant amount. Physiologically, actually, they very actually kind of important for us. Second, they friendly. Different kinds of probiotic commonly available as a supplement. You know, you can buy it in any store. And our hypothesis is that development of potential, for example, let's say radiation protective drugs or agent, which would produce in intestinal bacteria and protect astronauts from radiation will, of course, not require any payload. It will be with the people and will be produced. Of course, it's just kind of only idea, but let's just consider this because now it's very simple actually to design this, and our laboratory already make proposal for such kind of research when we just incorporate for example, one of the most potent radioprotective drug, which I mentioned probably in my previous presentation, flagellin-based radioprotector, is a just kind of fragment of flagellin which interact with Stol-5 receptor in our intestine and very significantly protect against radiation, boosting immunological response. And it's possible to incorporate, and we already now just design such kind of plasmid, when we incorporate this gene in plasmid and transfer to bacteria, to probiotic bacteria, which start to express and release of this gene or product protein to environment, and will immediately interact with the Tol-5 receptor in our intestine. Of course, it's very simple. It will be incorporated in a yogurt, let's say. If astronaut doesn't want, for example, this, they can take antibiotic and flush out of these probiotics. So second one, of course, project which we also believe very exciting actually, it's about actually ascorbic production. You know that humans lost during their evolution actually ascorbic production genes. Actually 4 genes, I guess, 1, 2, 3, 4, involved in synthesis of ascorbic acid in our organism. And we know that ascorbic acid has an important role in synthesis of collagen, for example, and it's also as a scavenger of many reactive oxygen species, important to protect tissue against oxidative stress and the chronic disease, including cardiovascular disease and even cancer. Well, and we know that ascorbic acid is why essential nutrient for humans because and non-human primates because they cannot produce this drug. And you know, it's very significant. For example, amount of these drugs which require. Idea is that this gene, which presented on the bottom of this slide, you can see which lost or damaged because due to deletion, significant deletions in primate and in guinea pig, it could be restored. And it could be also incorporated in plasma and produced in bacteria. In this case, we will have actually continuous delivery of ascorbic acid. And you know that human actually consumption vitamin C, it's approximately like 90 milligrams per day. And what's remarkable, if you compare, for example, mice, which has actually this gene and kind of produce in organism ascorbic acid. For example, plasma level of ascorbic acid with capacity, antioxidative capacity is 72%, versus in human, which is 0 or 20%, which coming, of course, from the food. And you know that goat, for example, if you scare this animal, its level of ascorbic acid in blood can increase 40 times, actually preserve this animal from oxidative stress. So we believe that it's one of the projects especially good for students which working in our laboratory when they incorporate this gene and transform to probiotic and check its production. So the next project, which also exciting, it's about again radiation, because we believe it's one of the major threat for life and astronaut. And we know also, I would like to probably to demonstrate that life on Earth always was under radiation. Even in early time, radiation level was much higher. And unfortunately, for some reason, and you know this reason, of course, we using only very limited spectrum of radiation as a source of life energy. And it looks like very unfair. Next slide, I just kind of demonstrate a different level of radiation which is acceptable for humans. Yeah, and the most important that a typical level is like 2.5 millisieverts per year, and you can see that what we have 250 millisieverts, it's already what we observe currently in Fukushima during this accident. And over this kind of level, it's already caused irreversible cancer development, which may be like in 2 and 3 years. Level of radiation, which we expect, like I mentioned in my previous presentation, during our trip to Mars, it's very high. And it's possible if we will not have any appropriate protection, it will be one-way ticket. So we just suggest that why would not we design of some bacteria which would utilize ionized radiation as a source of life energy. It's never happened. We have not such kind of example yet, because as we know, DNA and RNA is very sensitive actually to radiation. Of course, radiation is very important actually factor which actually drive evolution, producing some mutations and adaptation of organisms. This is why we have such kind of variety of organisms. But at the same time, you know, it's very strange that our life is based on genetic material which is very sensitive to radiation. At the same time, this much higher source of radiation which was in our planet in early life, nevertheless actually has such kind of positive contribution. Where is it a limit? At the same time, we know one thing. We're using limited actually visible sunlight for photosynthesis, right? Which convert actually CO2 in carbohydrates and glucose and oxygen. Only limited data actually indicate that ionized radiation can be still used as a source of life energy for organism. And this kind of maybe anecdotal, of course, evidence, but for example, we know that some bacteria which living on the bottom of ocean, photosynthetic bacteria, actually alkalies, they not have access to light. It's absolutely darkness. But nevertheless, they can be exposed to actually ionized radiation. For example, experiment with this bacteria demonstrate that continuous exposure to cobalt gamma rays, for example, without light, actually shows that these organisms start to live even better under this environment. And it's clear that mechanism of action of such kind of example when radiation can be used as a for photosynthesis, like in parentness, of course means that probably it's not similar to whatever we have in photosynthesis, which involve activation of chlorophyll, which require probably only many protons activated in signal actually way to reactions center to hydrolyze water and produce energy. So we believe that probably there is some kind of evidence, and one of that is radiotrophic fungi. I would like to bring it because this fungi, they call it mineralized fungi. Very similar structure, not similar, but still it's kind of pigment. Similar to chlorophyll, which presented on the right part of this slide. What is important that we believe that radiotrophic fungi may use pigment, this melanin, to convert this gamma and beta radiation to some another forms of energy. Why? Because you know Chernobyl. It's one of the kind of biggest disaster, and Fukushima is second example when people just not careful with radiation exposure. It's already 20 more years. We have big problems in Chernobyl area, and what people actually researchers found there, it's very remarkable. They found that probably 25 or even more, I believe that it's even more than 100 of general species of fungi could be isolated in this area. So last time they sent robot to the dorm, to Chernobyl reactor, which scratch from the walls of some samples. And what they found bacteria which perfectly survive in a actually very radioactive environment. And then last, I mean, on the corner, I showed some kind of trees in Chernobyl area. I know personally several researchers from Belarus which studied this effect. They have a lot of fungi which grow toward gradient of radiation. So it's thought that still melanin convert somehow ionizing or capable to convert ionizing radiation on another suitable energy which gives energy for life actually for life of this bacteria. What's important also that this melanin not so different from our skin. I remember many years ago in the Soviet Union it was actually very exciting several kind of experiments which they try incorporate of melanin to human skins. Of course, it's dangerous. What at that time experiment and idea was that it's probably will demonstrate some kind of protection. What we know also in Belarus, you can buy some kind of mushrooms, which is kind of fungi, in a pharmaceutical kind of store. And they believe that these mushrooms will protect against radiation. Of course, it's everything confusing at this moment, but we in our laboratory, just with scientists from the Albert Einstein Institute in Boston Medical School, Dr. Dashkova, we're trying to kind of probably to plan some kind of experiments which would demonstrate whether melanin capable to actually convert like chlorophyll ionizing radiation to another source of energy which could be used for different life forms. You understand that consequences of that would be dramatic. In this case, of course, you don't need probably some special condition if you have resistant bacteria to radiation to live on the surface of Moon or Mars. And that would be only initial stage, of course. I would like to present here just kind of last slide when students from IGEM team have some project which actually at NASA also, you know, running when they're trying to genetically modify E. coli bacteria to produce melanin. And you can see in the left picture, for example, normal Petri dishes with E. coli actually, and E. coli which produced melanin and black melanin. Of course, questions still remain whether this E. coli will be resistant to radiation. And another just kind of a story when oppositive, same students from, say, Australia in 2009 already start experiments when they incorporate chlorophyll in Escherichia coli and trying to actually to see whether it will be incorporated to metabolism to use some energy for as a source of energy for E. coli. All this experiment, of course, very exciting. Yeah, we believe that synthetic biology is kind of becomes a very important part of space research, bioresearch, because we can modify some terrestrial forms of life, make them kind of resistant and capable to live in under some certain environment in space, like in Martian circuits. And by this, we probably can start to colonize the planets. Thank you very much again. Yeah, I just would be happy to hear any questions.